Now let us move on to study the human excretory system. The human excretory system or the urinary system consists of a pair of kidneys, a pair of ureters, a urinary bladder, and the urethra. This structure that lies atop of the kidney is the adrenal gland. This large vein here is what we call the inferior vena cava, and this is a branch of the dorsal aorta. The projection of the blood vessels that you see here feeding each kidney are called the renal veins and the renal arteries. The kidneys are reddish brown, bean-shaped organs that are embedded in a mass of fat towards the back of the abdominal cavity. They can be found situated between the last thoracic and the third lumbar vertebrae, close to the dorsal inner wall on either side of the spine. And the average size and weight of the human kidney is 10 to 13 centimeters long, 5 to 7 centimeters wide, and 120 to 170 grams in weight. The kidneys lie just above the waistline, and the left kidney lies slightly higher than the right kidney. As you can see in the image here, from the kidneys, the urine moves into the ureter. The ureter is a narrow tube that connects the kidney to the urinary bladder. Urine from each kidney passes through the ureter and is taken to the urinary bladder. The urinary bladder is an elastic muscular bag which serves the function of storing urine. When the bladder reaches capacity, this urine needs to be excreted from the body, and this occurs through a narrow duct called as the urethra. The urethra is the duct through which urine passes out of the body. An interesting fact about the bladder is that here at the bottom of the bladder, there is a sphincter muscle which is used to control urination. When the bladder is full, the bladder sends a nerve impulse to the brain saying that Oh, I'm full now. I cannot take any more urine. This needs to be expelled. The brain will send a nerve impulse back to the sphincter muscle, which will cause it to relax, and now the urine can be removed from the body. But to some extent, we are also able to control the contracting and relaxing of the sphincter muscle. That is why, after a certain age, children are able to control their urge to urinate. The kidney is the site of urine production, and hence it's important for us to learn about its structure in detail. This region of the kidney is the surface that faces the vertebral column, and it is concave in nature. And somewhere here in the concave region, there is a depression at which the renal artery, renal vein, and nerves enter the kidney. And we call this depression as the hilum or the hilus. Inside the hilum, there is a broad funnel shaped space, referring to this space here. And here, there are some projections called as calyces. So these are the calyces, and this funnel shaped region is what we call as the renal pelvis. We can mainly divide the human kidney into two regions, the cortex and the medulla. The entire kidney is covered by a thick layer called as the renal capsule or the fibrous capsule. And inside of the capsule, we can divide the kidney into two major regions, the outer cortex and the inner medulla. The cortex is the outer dark red region, and it is covered and protected by the fibrous capsule. The medulla is the inner pale red region, and it contains the structures called as renal pyramids. These conical shaped masses can also be called as the medullary pyramids. The medullary pyramids project into the calyces of the renal pelvis. The regions of the cortex that separate the renal pyramids are called as columns of Bertini, or simply renal columns. The human kidney contains at least 12 to 16 renal pyramids. The radial stripes that are present on the medulla indicates the numerous kidney tubules. The kidney tubules or nephrons are the basic functional unit of the kidneys. This is where urine is formed. Just to recap, the basic functional unit of kidneys are the nephrons, which are present in the medullary pyramids, and they are visualized as these radial stripes. The medullary pyramids are present in the medulla region, which is the pale red region of the kidney, which is surrounded by the darker red cortex, and this dark red cortex is surrounded by a fibrous capsule. Now that we have covered the internal structure of a kidney, let us further zoom in to look at the fine structure of the nephron. The four main parts of a nephron are the Bowman's capsule, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Each human kidney contains about 1 million nephrons. Let us slowly break down the structure of a nephron and try to understand how this tiny but complex unit works. First, the Bowman's capsule. Each nephron begins in the cortex as a cup-like structure which we call as the Bowman's capsule. So, the Bowman's capsule is present in the cortex of the kidney. Then, the capsule leads into a short, convoluted tubule which straightens out as it passes into the medulla. So, the convoluted part of this tubule, right here, is what we call as the proximal convoluted tubule. As it stretches into the medulla, it straightens out and then it makes a U turn back into the cortex. This loop that is formed is called as the loop of Henle. So, this whole region is present in the medulla of the kidney, whereas this region is present in the cortex. Now, when this tubule re enters the cortex, it becomes convoluted again, as you can see here. We call this as the distal convoluted tubule. And finally, the nephrons, not just one but several nephrons together, open into a tube called as the collecting duct, which you can see here. So, several nephrons drain into one collecting duct. As you can see, the nephrons are surrounded by blood vessels, and these blood vessels supply the renal artery and renal vein. So, nephrons are surrounded by blood vessels. Blood enters the kidney via the renal artery, which branches out into arterioles. So, we have the renal artery, which branches out to form arterioles. Each arteriole will further branch out into a mass of capillaries. And these capillaries form a mass inside the Bowman's capsule, which is called as the glomerulus. The Bowman's capsule, including the glomerulus, can be called as the renal corpuscle. or the Malpighian corpuscle. So as you can see here, the arterioles enter the Bowman's capsule and turn into a bunch of small capillaries, a network of capillaries finely branched, and this network of capillaries along with the cup-like Bowman's capsule together is called as the renal corpuscle. Now, the blood that leaves the glomerulus will enter the blood capillaries surrounding the nephron, and they will unite to form venules, which in turn will join to form a branch of the renal vein. As you can see here, the blood is leaving, and then here the arterioles are forming venules, and together they come together to form a branch of the renal vein. Although it looks complicated, let us quickly recap the structure of the nephron. It is actually quite simple. So what do we have first? We have the Bowman's capsule plus the glomerulus, which we call as the renal corpuscle or the Malpighian corpuscle. And then we have the proximal convoluted tubule, and both of these are present in the cortex, which is followed by the loop of Henle, which is present in the medulla, and it takes a U-turn back up into the cortex to form the distal convoluted tubule, where the urine drains into the collecting duct. So this occurs in the cortex, and this occurs in the medulla. Some nephrons stay only in the cortical region, that is, the loop of Henle is not long enough, so it does not enter the medulla. And such nephrons are called as cortical nephrons. Whereas some nephrons have a long loop of Henle which runs deep into the medulla, and these, such as this one, are called as juxtamedullary nephrons. This network of capillaries that you can observe surrounding the loop of Henle, the network of capillaries that you see surrounding the nephron are called as peritubular capillaries. And in the case of the juxtamedullary nephrons, they form a U-shaped structure around the loop of Henle called as the vasa recta. With that, we have finished learning about the fine structure of the nephron, but we will now again further zoom in to the glomerulus and the Bowman's capsule to observe how urine is formed and concentrated. We know that excess minerals, waste products, and water needs to be expelled from our body. They are removed mainly in the form of urine. Urine formation takes place in the kidneys, and the blood leaving the kidneys usually has a lower concentration of the mineral salts, nitrogenous waste products, than the blood that enters the kidneys. So, what can we tell from that? We can tell that the kidneys act as sort of a filtration unit to purify the blood that is coming in so that the blood that goes out contains less toxic substances as well as less water. Urine formation in the nephrons consists of three major steps. First is glomerular filtration, which is followed by reabsorption, and finally secretion. Before we begin talking about glomerular filtration, this image here shows the ultrastructure of the Malpighian corpuscle. As you can see, the Bowman's capsule is a cup-shaped structure that is only one cell layer thick. And here you can easily visualize the network of capillaries that form the glomerulus, as well as the afferent arteriole, which is bringing in the incoming blood, and the afferent arteriole, which is containing the outgoing blood. Let us begin by discussing ultrafiltration. Ultrafiltration or glomerular filtration is the process of removal of small molecules from the blood. Removal of small molecules from blood. The blood that flows from the branches of the renal artery enters into the glomerulus in the form of these afferent arterioles. Most of the blood plasma gets forced out of the glomerulus and into the Bowman's capsule through this single celled layer. This process is called ultrafiltration for a reason. First, there is a high hydrostatic blood pressure that is present in this environment. The afferent arteriole, which is bringing blood into the glomerulus, has a wider diameter than the efferent arteriole, which carries the blood away, as you can see clearly in this diagram. Because of this, there is a pressure gradient that is created in the glomerulus, and this is the main pushing force required for the filtration process. Second, this single-celled layer of the Bowman's capsule is partially permeable. And since it surrounds this mass of blood capillaries, it acts as a very fine filter, and we call this as the basement membrane. It has very small pores. If we were to sort of zoom closely into this, if this is the surface of the capillaries, then we can say that the basement membrane is right here, and here you have these small pores through which small molecules can partially be filtered into the Bowman's capsule. So these small pores only allow water and very small molecules to pass through. Simply put, the high blood pressure that surrounds, that is present inside the capillaries, forces out the water and the small molecules for example, glucose, amino acids, mineral salts, and any nitrogenous waste products such as urea, ammonia, etc., into the Bowman's capsule. Let me just draw the basement membrane as well, so that we do not forget that there are two layers. And the material that is present inside the Bowman's capsule now, we call it as the filtrate, which is a suitable name because this is formed through a process of ultrafiltration. Larger molecules and cells such as blood cells, platelets, proteins and fats, they do not enter the filtrate because they are too large to enter the pores of the basement membrane. And these are retained inside the capillaries. So inside the capillaries you have blood cells, platelets, and macromolecules such as proteins and fats.